Hi, my name is Rashik. I'm one of the team leaders of the NYU AD iGEM team. And today, we'll be talking about synthetic biology, iGEM, and our project to 4 different people in 4 different levels of complexity. Through this exercise, we want to show that science is for everyone, and everyone should have the information that they need to make an informed choice. Join us as we talk about synthetic biology and explore this amazing field.
Hello, my name is Daulat Ali. I'm 17 years old and I'm a freshman at NYU Abu Dhabi. I'm really enthusiastic about biology and engineering.
So, let me start off with, have you ever heard of the term synthetic biology? What's like the first thing that comes into your mind when you think of synthetic biology?
I'd probably say that there are some materials that don't degrade. Like I've heard the terms like biodegradable, non-biodegradable, but nothing more than that.
All right. So synthetic biology is actually a really broad field. And since you said you like biology and engineering, this will be right up your avenue. So it encompasses a lot of different things. So everything from coding with your genes, that's a thing, yeah. And then actually editing your genome. So stuff like genome editing, that's synthetic biology.
Right.
And you can even have stuff like artificial meat that you make from like cells in a bioreactor. So there's really interesting applications for synthetic biology. And there's other functions such as actually making sheep that glow in the dark.
Oh, I
So
see.
you can do a lot with synthetic biology. And as I said, synthetic biology is like engineering of biology. So you use engineering principles to actually solve real world biological problems. So we use synthetic biology to kind of make systems that do not exist in nature, or kind of like improve in systems that are already in nature. So in essence, we're engineering them so that they do something that we want. Does that kind of make sense?
Yeah, it does. Totally does.
Amazing. And as I said, it exists right at the intersection of engineering and biology, which is why it's such a interdisciplinary field and has been gaining a lot of interest. Now, a bit about biology and how kind of our cells work in some sense. So have you heard of the central dogma of biology or know something
Actually,
about it?
I know this process.
Perfect. So you're already halfway there.
Yeah.
All right. So in essence, everything comes from the DNA. The information is stored in the DNA. So what we do in our project is actually give cells specific instruction to do what we want. So we're changing, in a sense, the DNA of bacteria.
I see.
Now, have you heard of collagen?
Yeah, collagen. It's a protein, right?
Yeah, exactly. It's a protein that makes up about like 30% of your entire body.
Uh-huh.
So it's a really big deal.
Yeah, it's mainly found in here, right?
Yeah, so it's found almost everywhere in your body. So you get from your eyes to your bones to your skin, and so it's ubiquitous, so present everywhere in your
I
body.
see.
Now, usually what collagen is sourced from is animals. So from like cows or like pigs or even like fishes. So what we're trying to do is produce collagen in bacteria.
I see.
So that's kind of the gist of how our project works. And so we're kind of like using cells as like mini factories.
All right.
So you can make cells do what you want by giving them specific instructions in the form of plasmids, which are basically just DNA.
I see.
And yeah, that's mainly about our project.
Oh, that's really exciting to find out more about it.
Amazing. I hope you get to experience the whole iGEM thing yourself, and hope it will be a interesting and a learning experience, as it has been for me, and I think a lot of my other team members would
Hopefully,
go the
hopefully.
same.
Hello, I'm Reem. I'm a junior at NYU Abu Dhabi and I'm majoring in bioengineering.
Hi Reem, it's really good to have you here.
It's good to have you too.
Perfect. So you said you're a bioengineer. So have you heard of the term synthetic biology before, and what sort of bell does it ring in your mind?
I mean, it brings quite a lot of buzz because I think that like bioengineering is based on synthetic biology, or it's a huge part of our major.
Uh-huh.
I think it's just like kind of integrating engineering and like physics principles with biology, or kind of like trying to alter like some concepts in biology or like kind of materials to kind of shift it to our advantage or kind of improve it.
Perfect. You could be sitting right here and explaining it to me. So, as you said, synthetic biology is at the intersection of engineering and biology. And so we're basically using engineering principles and applying them to real-world biological problems. And so that's where our project also comes in. So this year, we're focusing on biomanufacturing. So what we're trying to do is use cells, bacterial cells, as tiny machines for us, almost as factories. So what we're trying to do is we're trying to produce collagen from them.
From the bacteria.
Yeah. Have you heard of collagen?
Yeah, I mean, of course, it's a kind of produced by fibroblasts, which are basically skin cells, and it's also part of the extracellular membrane of the cell.
Exactly. So the extracellular matrix, and collagen is a huge structural protein which provides integrity to the ECM. Now, collagen, as you said, is synthesized by a lot of cells, and so it's present in our eyes, it's present in our bones, and a lot of different places. Now, how do we actually go about making a protein from scratch? Now, you can't shout at the bacteria and expect it to work for you. So how do you think you might actually make a bacteria produce something for you?
I'm actually not sure, but maybe you could like kind of target a specific. Not really sure. Enlighten me. It's
So
your area of
that's why
research.
you're here. So have you heard of plasmids?
Yeah, of course.
So plasmids are basically circular DNA, and DNA is basically information. Now you can actually code the DNA to produce what you want. So what we do is we literally put the gene for the collagen that we want. So we're using a bacterial collagen reg protein. So we get the gene sequence, send it to a company, they manufacture it for us, and then we put it in a plasmid, then put it in a bacteria.
Yeah.
Now, have you heard of transformation, bacterial transformation?
Yes, of course, yeah.
Exactly. So that's exactly what we're doing.
It's like a vector where you add a sequence.
Exactly. So we got the vector and we got our insert. We make the plasmid and then we use heat shock. So basically you kind of like make the membranes of the bacteria fluid, make it take up the plasmid, and then have it express it. So this also brings me to my next term, which is abstraction. Now, we don't really need to know exactly whatever goes on inside the bacteria. So we can give it the instruction and it can do all the work for us. Now, say if you wanted to go and make a protein from scratch, that would be a really difficult task. You'll have to put together amino acids one by one to actually get the final polypeptide. But with using a bacteria, you can get rid of all this trouble. It
Does
just
the
does
job
it for
for
you.
you.
Exactly.
Yeah.
Now, as you mentioned, there's a lot of different stuff that goes into actually making it and making sure that you have something that will produce the final desired outcome you want. Now, in this, there's like stuff like promoters where your RNA polymerase can bind, and then you get the final product through all of these various different steps. Yeah, and that's mainly about our project, and hope you had something informative from this very short exercise.
Yeah, like that's very smart. I mean, of course, we always try to get like shortcuts. If we can't produce protein, then why not let nature or bacteria produce it for us?
Exactly.
I think that's just a very good idea of integrating, again, engineering with and using their resources or even bacteria or other organisms to kind of reach a specific goal. So yeah, I think that's a very good idea and a very good project.
Yeah, and as you said, nature knows best.
Yeah.
And so sometimes there's this misconception that with synthetic biology, you're trying to do something unnatural, but usually we're trying to use nature in some way to get what we want in the end.
My name is Ibrahim Shihadi. I'm an instructor at NYU Abu Dhabi. I've joined NYU Abu Dhabi since 2012. I've been one of the PIs for iGEM ever since 2015, and it's been a great experience watching our students create and innovate and solve real-world problems at NYU Abu Dhabi.
Ibrahim, you've been a part of iGEM for 8 years at this point. What makes you keep coming back to iGEM, and is there a specific year or a memory that you associate very fondly with iGEM?
Um, I return to iGEM every year because of the energy we get from our students. The creativity, the input into the field of synthetic biology that students bring, the innovation that they bring into the project is absolutely incredible. The capabilities of our students at NYU Abu Dhabi has always surpassed expectations, and to be part of this process for them, to help watch them grow and mature as part of the iGEM program, has been an incredible opportunity for myself. One of my favorite years, I don't have a particular one, for example. They've all been special in their unique ways, but just seeing the students, rise up to the challenges that they face throughout iGEM, come together as a group, set up camp in the teaching lab, and bring their diverse backgrounds into the project, from biology, engineering, computer science, and all the aspects that come into the iGEM project, has been special from year to year.
That's incredible. And we're definitely super lucky to have you in the team as well. Now, you also teach a course on synthetic biology. What do you think is one of the most challenging topics that you come across while teaching the course?
With respect to synthetic biology, the biggest challenge is taking the theoretical concepts that we teach in the course into a practical world, and being able to switch the information from a theory to lab practice is a bit challenging for the students. And it's just about how to approach a course of such, how to simplify the lab experiments in order to be able to grasp the theory and the lab practice at the same time, while maintaining the ethical implications of the research, or the genre, which is the synthetic biology comes into play.
So people's perspective on synthetic biology can sometimes be quite negative. For instance, there's a lot of backlash when you talk about genetically modified organisms or even genetically modified crops. How do you feel about the discussion around the social perspective on synthetic biology?
From a social perspective, the fear that people have regarding the field of synthetic biology is a valid fear for everyone to have. However, just what's really important is the transparency, the ethical implications, and maintaining that all practices from the field of synthetic biology is done under an ethical and regulatory practices. Then it has a huge potential to benefit the society and the field of biology in general.
Yeah, I definitely agree. It's a very important conversation to have. And I also fear that sometimes there's a lot of misinformation regarding synthetic biology. And actually having a platform like iGEM, where we have stuff like human practices, where you actually go and talk to people, let them know about what synthetic biology is and the benefits of it as well. It's a very important step. And in order to do that, we actually did a tabling event on campus, where we went and talked to other undergrads on campus, and we got their input on our project, which was the production of vegan or halal collagen. So collagen usually is derived from animal sources, so like cows, pigs, and sometimes that can also have like a religious implication. So what we wanted to learn was, do people actually care about it? And based on the responses that we got from the survey, what we actually found was that people actually don't know a lot. So they don't know where the collagen products that they use are coming from. And so what we want to do is make that transparency, as you mentioned, so that they know that they're getting a safe product that is ethically sourced and that they can trust in.
That's amazing. You guys are doing a great job.
Okay, hi everybody. My name is Rafael Song. I'm associate professor in bioengineering at NIAD. I'm also head of bioengineering program since 2020. I've been involved with iGEM since 2017, and now I think the topic this year will be on biomanufacturing of collagen, which is quite exciting, and I'm really looking forward to seeing results coming from the team, and hopefully we can win the grand prize at the Jamboree in Paris.
That's the hope. That's the hope for sure. And as you mentioned, you're the head of the bioengineering program as well at NYU AD.
Yes.
And then how do you think iGEM fits and the whole synthetic biology fits into the bioengineering program?
Right. I think synthetic biology is really the new emerging field in bioengineering. So I'm really glad that students take this initiative and work on synthetic biology in this extracurricular activity. So I believe synthetic biology will be a very important field in the future area of bioengineering. So I think I believe there's a really good fit between synthetic biology done by iGEM and the future of bioengineering.
That's incredible. Now, what we'll do next is just run through our project and the science behind it in a bit. And we just want to hear your feedback and what do you think about the project in general.
Yeah.
So what we are targeting this year is specifically at producing bacterial collagen-like protein. Now, bacterial collagen-like protein differs from human collagen in that it cannot form fibrillar structure. Now, this is because it lacks a very important post-translational modification, which is hydroxyproline. Now, that is because bacteria do not have the enzymes to produce, to actually do the hydroxyprolination. Now, what we're trying to do with our project is actually force the bacteria in some way to actually take up the hydroxyproline, which we'll get to in a bit. Now, how we're actually doing that is first making a plasmid that contains our gene of interest. So we got the gene from an actual bacteria, we put it in our vector, and then we hope we put it in the bacteria and then make the bacteria work as mini factories for our cells. So what we do next is something called gene code expansion, where we actually do the incorporation of non-coding amino acids or unnatural amino acids. In our case, that would be through the hydroxyproline and hydroxylysine. Now, there's two main approaches that we considered for our project, and the first one being using a tRNA synthetase enzyme. But the problem that we faced with that is we couldn't find appropriate tRNA synthetase system that we could get in time for our project. So what we do next is, as I said, kind of force the bacteria to take up the hydroxyproline. So we grow the bacteria in an amino acid medium that lacks proline, but then we add hydroxyproline. So the bacteria has kind of like no choice but to grow with the hydroxyproline. Now, an important thing is here that the bacteria cannot be able to synthesize the proline themselves. So we use a special auxotrop strain, and so that kind of prevents the proline from being synthesized, leading to the incorporation of hydroxyproline. And then the incorporation of hydroxyproline leads to the fibrillar formation and gives the desired properties that we want. And once we have that, we basically just purify it using NINTA system. So it's like an affinity chromatography. And then the intended application could be anything from cosmetics, where you use it for like dermal filters, or even like cosmetic creams and stuff. So that's where we're at with our project so far. And hopefully we'll get the collagen soon, do more analysis, and have it by the Jamboree.
Great. I think this is a really great topic. I do believe that this is a huge potential. I believe because it's not just for cosmetics. I think this can be also used for biomedical applications. If you have to use some tissue engineering approach to grow collagen, I think this could be really, really, scalable and low-cost manufacturing method. And I think this has also big potential in bioengineering and biomedical engineering as well.
Exactly, and even outside of iGEM, as someone interested in tissue engineering and regenerative medicine, having like a suitable collagen source is super important.
Right.
So right now, in one of the projects that we have in the lab, we're trying to get collagen from like fish, and that takes a very long time. And you kind of start with like a fish smoothie kind of thing, which is not very pleasant to work with. So this could be like a suitable alternative to that.
Right. Yeah. Now, how easy is this to extract collagen from bacteria? Have you sort of how easy and how expensive is that method compared to a traditional method?
Yeah. So we looked at how synthetic biology will change the whole biomanufacturing scenario. So there was this report by BCG that said that in the coming decade, synthetic biology could account for almost 50% of global manufacturing. And that's like a huge number, something like $20 trillion.
Wow.
So there's a huge potential for scaling up these systems, and even for things like insulin, where we already see that this approach is already there. So I think one of the challenges with collagen specifically is when you try animal collagen, it takes a lot more effort. So this could be like almost like a blank canvas. So you can put in various features that you want into the gene itself to get the desired property. So this allows for a great deal of tunability as well, which we hope to somewhat focus on in our project. So Professor, could you please tell about how synthetic biology kind of like incorporates with your own research that you do at NYU AD?
Okay, I am working on various topics in my research, and one of the main pillars is on biosensing. So I'm very interested in developing a biosensor that can be used to detect various pathogens and diseases early on. So I do think the synthetic biology can offer us a new method to detect those pathogens and disease biomarkers early on. And that's sort of my personal take on the future of synthetic biology, that this can be also used for detection of various diseases and pathogens, and this could help to improve the health of humanity.
That's incredible, and we hope that's a future we get to see very soon.
Yeah.
And thank you so much for listening in with our conversation with Professor Song, and stay tuned for more updates on our project. Thank you, Professor.
Thank you, guys.